The migration and differentiation of neural crest cells is influenced by their environment, and that environment changes along the anterior-posterior axis. Your textbook uses the analogy of traffic to illustrate this point. We'll see that some types of neural crest cells, like trunk neural crest cells, migrate preferentially individually, and this is particularly true for those cells that give rise to pigment cells or melanocytes. Other types of neural crest cells, like cranial neural crest cells, migrate in clusters, so they exhibit what is known as collective migration. Another aspect that is very important for the migration of neural crest cells is the presence of guidance cues. These can be long-range or local guidance cues that are important for guiding neural crest cells along the way. In addition to that, physical barriers will also affect the tissues in which neural crest cells can be found. Neural crest cells first become apparent when they undergo an epithelial to mesenchymal transition, and in the context of the neural crest, this EMT is known as the lamination. Recall that when we talked about gastrulation, we said that the lamination referred to the splitting of one cellular sheet into two more or less parallel sheets. But in the context of the neural crest, the word delamination simply refers to the separation of the neural crest from the neural epithelium. For most cells, that means a process of EMT, that is the separation of these epithelial cells into individual mesenchymal cells. But for some neural crest cells, that process involves low levels of adhesion that will keep the cells as clusters, so they are loosely associated with each other during their migration, as we'll see later. Now, this timing of the lamination is controlled by the levels of BMP. Recall that there's BMP that's secreted from the dorsal aspect of the neural tube, but there are also inhibitors like nuggin that are produced by the notochord and by somites, which are located around this region, and these somites are going to be giving rise to the vertebra and muscles of the back. And while these nuggin and other BMP inhibitors are being produced, the neural crest cells cannot undergo EMT. So when nuggin expression is reduced, the BMPs can function and activate EMT in the neural crest cells. Just like the separation of the neural tube from the overlying epidermal ectoderm requires changes in coherent expression, so does the delamination of the neural crest cells from the dorsal aspect of the neural tube. As you guys know, the neural tube expresses N-cadherin and the overlying ectoderm expresses E-cadherin. Now, the neural crest cells express cadherin 6B right before their delamination, but in order for them to leave the neural tube, they actually have to bring down their expression of cadherin 6B. And this is made possible by BMP and Wnt, which lead to the expression of the transcription factor SNAIL, which is going to promote the downregulation of cadherin 6B and in turn promote the epithelial to mesenchymal transition of the neural crest cells. Now, it's important to note that before the neural crest cells express cadherin 6B, they actually express N-cadherin since they were part of the neural tube at one point or in the region of the neural tube. Another important thing to note is that there is a subset of neural crest cells, specifically the cranial neural crest cells, that will express N-cadherin once again when they begin migrating, and that will allow them, as we'll see later, to migrate as clusters, so to stay together. And they express it at low levels. Finally, another important aspect to note from this figure is that the movement of the neural crest cells is going to involve changes to the actin cytoskeleton that are mediated by the Wnt non-canonical signaling pathway, which is the planar cell polarity pathway. In order for neural crest cells to leave the neural tube, so to be pushed out of the neural tube, they undergo a process that's known as contact inhibition of locomotion. And that simply means or refers to a phenomenon by which the neural crest cells change their protrusions of the cell membrane so that the protrusions face away from a side of a point of contact with another neural crest cells. So wherever these cells make contact with each other, they cannot form protrusions to migrate forward. So their migration is going to occur in the point that's away from the side of contact with another cell. The process of contact inhibition of locomotion involves the activation of the non-canonical Wnt pathway or the planar cell polarity pathway. And this pathway is going to be activated when neural crest cells make contact with each other but not with other cell types. Now, the Wnt planar cell polarity pathway is going to activate a protein Rho-A, which is going to change the actin cytoskeleton to basically desegregate lamellipodia, which are these protrusions that form in the plasma membrane as a result of the polymerization of actin filaments inside of the cell. So these lamellipodia will desegregate at the point of contact, and they will form at the site that is away from the point of contact. Now you can imagine that that would lead to the dispersal of cells. And that's important for the migration of individual cells, as it's the case of the trunk neurocrest cells, particularly those that give rise to pigment cells, which usually migrate on their own. But this is also important for the migration of cells that move as clusters, because when cells are moving close to each other, they are in contact with other cells all around them. So the protrusions do not form around those point of contacts, but only form at the leading edge of a stream of cells. And this is what is shown in this image, which shows a pattern of migration known as collective migration, which is exhibited by cranial neurocrest cells. And this pattern of migration that is maintained by this group of cells is made possible as a result of ongoing contact inhibition of locomotion among the cells at the leading edge. In addition to contact inhibition of locomotion, collective migration also requires that the cells express low levels of N-cadherin, which are shown here as the blue dashes. So this N-cadherin expression at low levels is needed for optimal collective migration. And if you increase the levels of N-cadherin, that would prevent the cells from being able to invade spaces. So as you'll see in a video that I'll show you shortly, the presence of that small amount of N-cadherin actually allows these cells to behave almost like a microfluid when they encounter a narrow space. In addition to expressing that low level of N-cadherin, the cells also secrete a factor which is known as C3A, and is a coattractant. So this factor allows the cells to be attracted to each other, and that helps them to migrate together. Now, this process of collective migration is considered to be an intrinsic property because it's seen both in vivo and in vitro, and it doesn't require external cues. So what that means is that it doesn't require chemoattractants that are secreted by cells other than the neurocrest. However, the neurocrest themselves are secreting the coattractant C3A to remain as a cluster. These movies display the process of collective cell migration in cranial neurocrest cells in vitro, both under normal conditions and also under conditions at which the levels of N-cadherin are higher than usual. So from these movies, you can see that the low levels of expression of N-cadherin, while allowing the cells to remain as a cluster, also gives them the ability to have a solid to fluid-like transition so that they can squeeze through small spaces, like the spaces that they will encounter during their migration in vivo.